into the specific muscle they are supposed to. So what will happen is that, this is the same as our, finger or something. So let's say certain neurons, motor neuron comes through the your limb, and then they supposed to certain neurons supposed to touch the thumb or something so that you can move this thumb. But let's say how about that neuron kind of lost, and then what if they innovate to a different finger? Then what will happen is that you try to move the thumb or something, and then end up you are moving the different fingers or something, isn't it? Right. So this is like sometimes it can be very, very it causes very, very significant consequences. What I heard is that we don't have those figures in here, but some of the motor neurons that coming from the brain is cross the side, and then so something coming from the right is controlling something on the left. But let's say that is somehow their navigation or migration is somehow messed up, so that let's say they don't cross the midline, and then end up you try to move the left, but end up you are moving the right hand side. So that kind of consequences we can get if this cell migration is somehow defective. So I'll jump to the some biochemistry here, okay, in the next slide. So if you have any question, feel free to stop me so that I can answer your question. So this is the what we call guidance cue. So all this movement, okay, all this cell migration is that, it was in the in our even midterm exam, isn't it? The 3 requirements for the cell migration, and you need to have an extracellular matrix, rough surface, and then the motor, which is the actin cytoskeleton, and then third one was the chemoattractant, mostly like it's a chemotactic agent or chemical. So that is the chemical sensing. Think about it. Few slides back, I talked about growth cone, but growth cone does not have eye, right? So only thing that they can really sense the direction is the biochemical, and then probably they are following some kind of chemical gradient. Okay, right. So based on that kind of property, and then using the also biochemical property of this isolated some of the suspicious molecule, and then what they found is that, okay, largely there's a 2 signal. So that is green light and then red light. Okay, so green light is the go signal, kind of like attractive. So come up here. Okay, so you are attracted by certain chemical. So that's why you are following through that chemical. So you are trying to go to the area that the concentration is very, very high. Okay, right. And then the other one is, and you look at it, is the red signal is that repelling signal, isn't it? So it's very kind of like it's easy concept. But in biochemically, it's very kind of difficult. Why it's difficult is that, okay, growth cone has the receptor, okay, but they have to sense those chemical, and then they'll repel it, isn't it? So they'll move away from it. Okay, so it's kind of like this cell has a receptor, but kind of like I said, they hate this chemical or something, so that's why they are moving away. Okay, right. So that is kind of like 2 different type of concept, attracting and then repelling, and that really determines the go and stop signal. And then another is the quality of the signal is that another one is long range signals. Okay, cues. They call it a long range cue. So it's a long distance. So mostly these long range cues are secreted produced from the cell. It could be from the cells and the surrounding of this neuron, or neuron itself may produce some kind of cues. But that is the long-range cue, so it is secreted outside the cell and deposited in the extracellular matrix, most likely. And then, they forms the gradient. And then, but that can be also, attractive or repellent. And on the right is the short-range cue is the, it's not really, I mean, I think it is the secreted, it could be a secreted molecule, but most likely that is deposited on the surface of cell, okay? So that's why we call it short-range cue. And then let me write it down in here. One in here is that, okay, that is contact dependent, okay? So how the receptor and then ligand is really meeting is that it's not by the, I mean, ligand and then receptor still form a complex, but what will happen is that the migrating cells somehow touch the cell surface, and then they will know about it. Okay, so that is the attractive, then they will stay, and then they'll go to the next one. So that is the I would say contact dependent. So cell to cell contact. Okay, let me cell contact dependent. Okay, right. Okay, so so it could be green signal, isn't it? But if they don't like each other, then they'll just move away. So it could be the also repelling signal also. Okay, so largely we have this kind of signal. So it could be a chemoattractant or chemorepulsion, but it could be a long-range cue. But also it could be a contact attraction or a contact repulsion. But it could be a green signal or red signal, which is the repelling, attracting or repelling. Okay, so next. Sorry.
Dr. Lee?
Yeah. Go ahead.
How do we, what's considered a long range and what's considered short range for cells?
Okay, so long range is something that's secreted out, the ligand that is produced by cell, and basically, let's say, extracellular matrix long range cue, isn't it? Right, so that is secreted outside. And then the short range cue is that something that expressed on the cell surface. When they bump into each other, and then they will know whether that is attractive or repulsive. So that's why they call it a short range cue. Okay, good. Okay. Okay, so this is the look at this. This table I copied from 2001 paper, and I think that's we don't really have any addition here so far. Okay, so this is still basic. So these are the cues that they found. We're going to talk about netrine in a while, but before the netrine, discovery, I think this kind of cue was largely not very well characterized. So I think that back then, 2001 was very recent information, and I think that's I haven't heard anything that addition to this. I think there are some other molecule people find, but I don't think that's we can talk about it in like a class. Okay, I think that is more like still research level. So what they found is that these are the ligand. Okay, so attractant, also repulsive cue or attractive cue. They are all, we can call it as a ligand. Okay, so that is the cue. And then receptor is the molecule that is expressed on the migrating cell, surface of migrating cell. So these are the receptors. Okay, right. So here, so these are the receptors. So let's say they found netrine. Okay, we're gonna talk about netrine I think we're going to talk about everything in here. But the natrin is their ligand. Then they find out there are two kinds of receptor binding to natrin. So these are the receptors. So there's ONC40, or in vertebrate, it is DCC, and then ONC5, and in vertebrate still they call it ONC5. But look at this response is that, okay, when they have the cell have a DCC, then response to the natrin is attraction. So if cell is only expressing ONC40, then ONC40 is the attraction. So they attracted to the natrin. But the certain cell is expressing ONC5, then what will happen is that basically they will move away from them. Okay, so repulsive signal. So I think that this response is determined by what kind of receptors they have. Is probably an important thing. Okay, so and then slit, we are going to talk about that in the Drosophila neuron, and they have a robo is their receptor, and this is the robo is the what is it, roundabout, okay, and they have a some kind of repulsive or adhesive consequences in here. So, and then the afferent is afferent, and then afferent receptors are most largely repulsive. And I think the semaphorin is also pretty much repulsive. Very minor cases, they can be attractive signal. Okay, so bottom line is what it is that same receptor and then the same ligand attraction, but depending on cell, I would say depending on tissue or depending on context, it could be a different consequences. So sometimes it can be a attractive signal, and sometimes it can be a repulsive signal. That is really depending on the cell and then tissues, sometimes by the organism. So what we're gonna learn is mostly about the kind of like a principle, kind of like a general information. Okay, right. Okay, so next is that the those kind of guidance cue that I listed in there. See, look at this. It's not only important for neuronal cell migration, but they are also important for mesoderm cell migration. And semaphorin is also what we're gonna see is the neuronal cell migration example that we are going to see is the repulsive signal. But actually, this is the bone and heart morphogenesis. The semaphorin is very, very important also. And then afferent is the neuronal cell and the neural crest cell migration. And then also important for this angiogenesis, blood vessel formation. They are also important. So I think this slide tried to stress these ligands not only for the migration, but they can be also used for other functions during the development. Okay, right. Okay, so from now on, I'm going to go over just basically one by one. Okay, right. So next is the semaphorin. Okay, so semaphorin is I think I have if you see this note in here, and I added pretty much detailed information. Something that I want to talk about in here is in here. So I think I already put like a narration in here. So if you want to know, in detail, or if I miss something, or if you miss something, probably you might want to take a look at this note. So basically, semaphorin is primarily is a repulsive cue. So this is the developing limb. So usually limb, we have a limb here. So you have an arm and you have an arm and you have a leg, right? So this is like we consider development of limb is the post-embryonic, because think about it, all our body is composed of 2 layers. So ectoderm, and then mesoderm, and then the endoderm, right? But the limb does not have endoderm. I mean, we don't have intestine run through this limb, right? Right. So it only has an ectoderm and then mesoderm. Okay, right. I think so. That's why they have the I kind of like added the what is it axis in here. So this is a limb. Okay, I think dorsal is the your, let's say we'll put it like this, and the dorsal is the knuckle side is dorsal. Okay, and then I think that this is the ventral is the palm side. That is is easy in this what is it, the hand, but I think that entire arm has the dorsal, and then I think that where your elbow is, that is the dorsal. Okay, and then the other side is ventral. I think that's how going that way. Okay, right. So what it is that in here was it TI1 neuron that is in a developing limb, and then neuronal cell body is here. Okay, and then so I'll just move to a arrow here. So here, and then they are stretching axon, isn't it? Okay, and then they'll hit the semaphorin band. So because of that, semaphorin is a recursive cue, stop, right? So they'll stop right there. But the thing is that they have to leave, they have to go. Okay, they still have to cross it. So they are exploring is there anywhere they can jump over this band? Okay, right. So they will usually run through this dorsally, right? But somehow, because of that, they change their position, and then they are going into the ventral side because of the CX1 cell. So that is the contact dependent. And then the CX1 cell is expressing something that the attractive signal. Okay, so CX1 is kind of like a guidepost cell. So here, okay, so they are giving the signal that, okay, you cannot cross that, but I have the, can wait to, so this cell is basically, producing some kind of attractive cue. So when this TI1 neuron hit this area, so they are coming down to this distal area, distal end of this ventral side, then they'll cross it. Okay, right. And then they'll keep on migrating. So consequence is that the this recursive cue used for changing the direction of this cell migration. So usually they run through the dorsally, and then at a certain point, and then cross this side, and then run through the ventrally, and then probably they can go into the CNS. So CNS is basically like a spinal cord. Okay, right. So although semaphorin is the recursive cue, but in this case, cells kind of like stop and then try to explore the points that they can cross, and then they can cross this band using the after they recognize CX1 cell. So one thing I want to add is that this kind of neuron, you have a cell body right here, and then they are stretching axon to the CNS, and then you see growth cone over here. That is, we'll say, sensory neurons mostly, isn't it? So they are sensing something, right? And then they are, delivering those information to the spinal cord. And I'm sure that maybe they can just make a turn and then go to the brain all the way. But anyway, this is like feeding it into a CNS, and then they try to make the uniform migration pattern or something. Then sometimes they have to change the position and then form a bundle over here. So that's why they will use this kind of recursive band, and then that gives the positional information, so they can change the direction and then cross in the only one place. So semaphorin, is really the recursive signal. I think this is the just experimental demonstration that semaphorin is the really recursive. So you have the some kind of sponge, maybe a cotton swab or like a cell that expressing semaphorin, and then you put the nerve explant, and then if you stimulate them, you can see nerve doesn't grow this way. Okay, this is a control cell not expressing semaphorin, and then they can stretch the axon this way. Okay. All right. So can I move to the next one? Okay. Okay. So this is the YouTube video. So it has the you know, what is it? You know, was a volume, you know, the sound in it. You might want to turn down the volume. Okay. So I'm gonna play. See if it works.
The human brain is composed of a network of billions of neuronal connections.
Can you hear it?
This network enables us to move, communicate, and remember.
Okay, good.
Its formation starts with the birth of neurons and their migration to the brain region where they will function. After reaching this region, neurons form two types of extensions, dendrites and axons. Dendrites are tree-like structures that receive synaptic inputs from thousands of other neurons. While neurons can generate multiple dendrites, they mostly form only one axon. Axons carry information from the neuron to other cells located in or outside the brain. Axons often grow over a long distance to contact their target cells. Their path is determined by guidance proteins. Guidance proteins either repel or attract growing axons. They're detected by a hand-like structure at the axon's leading tip, the growth cone. Different proteins act together to guide each individual axon to its final target. During a large part of its journey, an axon may converge with other axons into large bundles. Analogous to a car changing lanes on the highway, the position of an axon within a bundle changes during its journey. This position is regulated by guidance proteins present in the bundle and determines which part of a specific brain region the axon will enter.
I'll stop it. Okay. So this is pretty good information. Only like 2 minutes or 3 minutes is worth watching it. You know, it gives you an idea about, you know, axon and then growth cone and then, you know, attractive cue and repulsive cue, and then even the axon, you know, bundling, and then moving direction, moving the side, you know, changing the direction, migration direction. That is also contained in here. You know, it's a pretty good information. I strongly recommend you, you know, watching this, you know, YouTube video linked to here. So next is the about, you know, chemo. You know, one of the, you know, very well characterized, you know, chemo attractive or chemo repulsive cue. So I have the also big, you know, writing, you know, note in here. So let me share the different file. Okay. So before we understand, we need to know the what is the commissure neurons. What do we mean by the commissure neuron, and then what it is. So I kind of like try to find some better picture here. So okay. So do you see this file here? Isn't it? Do you see this spinal cord JPEG? Isn't it? And then you see it's a vertebrate spinal cord. This is not my drawing, okay? I just copied somewhere, okay? Right. So the reason why I have this is that, okay, so okay, let me change to you know, here. So look at this neuron here. So one of the neurons, so this is the, I'm gonna write it, okay? Here is the dorsal, so D is the dorsal of the neural tube. And then here's the ventral, okay? So the ventral in the center here, the red area, that is the midline. Okay, let me change, is the midline, or here floor plate, right? So we call it a midline or floor plate. So you can say the same thing in here also. Okay, so in here also you can say, hey, this is the dorsal midline, okay, right. Or I think that's they call it dorsal plate, okay, right. So that is the same here, okay. So you can see, let me just kind of like try to paint it, okay. So here, okay. So there is a rule, okay. What I want to say is that, okay, there is a certain rule is that, okay, the neurons, the commission neurons, the commission neurons by definition is, you know, crossing this, you know, side. So one neuron that started here, isn't it? Right, isn't it? And then this guy migrate to the bottom, right, ventral, and then they have to cross it. So they usually they are on here. Here is what I would say. Let's say here's the anterior, then this is a left, okay, and then let's say this is the right, okay, right. Then they they started over here, okay, and then they will migrate. Okay, they will migrate down here, right, and then they'll cross to the right. So that was in the left, isn't it? And then they are going down to the ventral, right? And then they'll migrate to the right, and then they'll move to the you know, wherever the direction is. So let's say here is the posterior side. Okay. So think about it. Let's say this is the brain. Then one neuron was on the left, somehow went down to the floor plate, and then cross it somewhere. Okay, probably ventral this way, right? And then they are migrating to the right. Okay, right. So probably that is the representing one of the you know, commission neurons. So commission neuron is by definition always neurons, somehow neurons that they are changing the direction during this axon formation. So that is the you know, definition of those. Okay, right. So the so rule number one is that, okay, so here, so in the midline, okay, the rule number one is that, okay, neurons can cross the midline, but cannot stay. Okay, so I'll just select it. Okay, so rule number one is that in the midline, so rule of midline is that neurons, okay, come down to the midline, but they have to cross it, but they cannot stay there. Okay, it's kind of like a, you know, is it the so they can cross it, but they cannot stay there and then migrate. Okay, so that is the number one rule. Okay, right. So I think that's, you know, okay, I'm gonna save this so that you guys can have as a you know, as a screenshot. So you know, number one rule is they cannot stay and then they have to cross. I think that is the really important information to understand this, you know, what I'm talking about with the net train. Okay, so I'm gonna move back to the PowerPoint. Okay. Okay, so I came back here. Do you see PowerPoint here? Do you see it? Okay, right. So did I make a mistake? Okay, I think I'm all right. Good. Okay. So here, so this is the you know, those you know, neural tube, and then see in here is a roof plate, right? It's a dorsal, right, and the ventral is a floor plate. So this is the midline. Okay, and then it is showing the commission neuron, and then commission neurons comes down here, and then they will cross it. So red line is a crossing, and then in this case, I put it as a, let's assume that they are migrating to this anterior side. Okay, so this is one of the you know, commission neuron. So they try to find, you know, how this commission neuron find their right path. Okay, so how they did is that, okay, so you know, the guy named I think that's Mark Tessier Levine or something, and then those people, what they did is that, okay, so I'm going to use this box here. Okay, So using this box is the something that you you know, what is it? You cut out. Okay. So using the I mean, with a carpet knife or something, you cut this chicken embryo spinal cord, and then somehow you take out the dorsal side. Then what will happen is that if you do that very early, then you have all this commission neuron cell bodies right here, isn't it? Right. So that is this. Okay, right. So what he did is that, okay, so so you cut out, and the cutout is right here. This is the kind of like explant of dorsal neural tube, and then one on here is that, okay, he also cut out this part. Okay, because all the neuron is coming down here, right? So what he did is that, okay, so he cut out this part, and then he isolated chemicals. Okay, he isolated chemicals. Okay, and then he isolate, using it's HPLC or whatever. Okay, and then what he did is that, okay, and then he just put those each chemical one by one on this here. So red, was it the orange block? Let's say this is like a either cell or either or is it the sponge or something. So it contains those chemicals. Okay. So let's say from this experiment, you are able to isolate like 10 different chemicals. And then they did an experiment, 10 different experiments like this, and then try to find whether chemical is causing this kind of activity. Think about it. You have the chemical here. You put a sponge here. So that's why, let's say, in this chemical has the attractive activity, then all the neurons migrate, and then also their axon will be stretched to the this side. Isn't it? Because they are attractive. Okay, right. So in this case, let's say you have a number 1 to number 10 chemicals. Okay, let's say this one was a number 9. Then hey, number 9 is the is the attractive activity. So that's why you can identify the chemicals that are responsible for this kind of activity here. Okay. So do you guys understand? Do you guys have any question?
I'm sorry, I got a little lost when you were saying the number 9.
Oh, okay. Number 9 is okay. So do you understand then the chemicals, this part?
Yeah, I understand.
Yeah, you take it out.
The chemicals are put into that cutout. With,
right. So let's say you from this experiment you isolated so many, the 10 different molecules. Let's say, for example, I just made up some kind of your experiment. Okay. So you are able to isolate 10 different chemicals from here.
Yeah.
Okay, right. And then you try one by one. So you have like a 10 different experiment you did with this kind of you did a 10 different experiments like this. Okay. And then let's say we'll just choose, if you don't like the number 9, then let's choose number 7. Okay. So number 7 chemical has causing this kind of activity. Then number 7 chemical is the attractant.
Okay.
Isn't it? Okay. Okay, got it. Right. So that's how they are able to find those netrin over here. Okay. Just a second. I have to. Okay. So here. So they found a netrin, and then it is from the came from numerous chicken. Okay. They isolated from chicken embryo. And the spinal cord, right? And then, okay, so I'll just remove this. This is a bug that it shows up in the next one, right? So it turned out to be netrin is very similar to laminin, which is the extracellular matrix molecule. But netrin is not laminin. Okay, so happened to be after isolated, it kind of a structure is one of the laminin here. I think laminin gamma chain or something like that. So it's a netrin that they found. So that's why they kind of like named it as a netrin. and then it is something that similar to laminin. And then so next figure, they look at the gradient of the this natrin, and they are more in the ventral. So can we label this? Okay, so here is the ventral. Okay. Okay, and then here is the dorsal. Okay, right. So I think, yeah, here's the dorsal side and here's the ventral side. Okay, right. So what it is that the this is the also panel A. Also, can we label this is a protein gradient? Okay, this is the protein gradient. Okay, okay, and in here this is an mRNA blot. So gradient is the what? They are ventrally very, very high. So highest in this floor plate with the ventral midline, and then as they go through the dorsally, they'll have a less and less amount of natrin. So probably in the dorsal plate have no natrin at all. Okay, there's no natrin at all. Okay, and then the in panel B they did they look for mRNA of the natrin. So what cells are producing this natrin, and then they were able to identify natrin is produced from the cells in this ventral midline, isn't it? Right. So cells in the ventral midline or floor plate cell. I don't think that we can really say ventral midline is a little bit overexposed. Okay. So, but anyway, the ventralmost cells are producing this, natrin, proteins. Okay. So let me go over one more, okay? And then, okay. So this natrin really have the chemoattractant ability, and this is showing the time course experiment. So as you can see, this is the one of the natrin attracting neuron nerve end, and it's a 0.15 minute, 45 minute, and then 60 minutes. Let me erase this, guys. Okay, right. And then the as you can see here, the black arrow is the pipette tip that's secreting purified natrin. Okay, so they keep it in the same with the photo frame, and then as you can see, natrin is moving toward the the nerve is moving toward the natrin sources, isn't it? So that is the demonstration that hey, this is really the chemical attractant. Okay, so now it's 12:05. So do you guys have any question?
No, so far.
Go ahead. Ryan, go ahead.
I was gonna ask how the participation points would work with the class.
Participation class.
Uh-huh.
So that is the, I don't have participation grade for the lecture.
Okay.
So are you talking about the lab?
Yeah, yeah.
Yeah, that is so I mean, I just don't have a really a good answer for that. Can you wait for me until I really figure it out?
Yeah, of course.
Yeah, I'm sorry. It's just I've been thinking about it, and I just couldn't find a better way to handle those. Okay, so maybe if you give me like a 1 or 2 more days, then I may have some better idea.
You can take your time. It's all good.
Okay, sure. Thank you. Any other question?
Dr. Lee?
Yeah.
On the slide before, when you showed the Netrin-1 mRNA in the floor line being produced there, you said that we couldn't say ventral line. Is that right?
Pardon me?
Did you say that we couldn't say ventral line? You were really particular about the wording there, and I was just curious why.
Oh, right. So ventral, they cannot stay in the ventral, midline. So think
in
about
the ventral midline, yeah, but not like the ventral line.
Yes, this is a ventral midline.
Okay.
Right. Okay. So in the ventral midline, the cell can cross it from here. Let's say here's the left. Okay.
Yeah.
And then they come down, and then they can cross it. They can go to the left, or some one in the right, and then they can cross it, but they cannot stay and then migrate.
Yeah. Okay.
Yeah,
Thank you.
but the thing is that, good question is, these cells that migrating down, they all have a natrin receptor, isn't it? These two cells. Then let's say if there's a signal is the attracting signal is only one signal available is natrin, then they will all come down to the ventral midline and then they will stop there, isn't it?
Yes.
Yeah. So that is the kind of like always a paradox, isn't it? So they come down here to the highest area of ventral midline with a natrin, right? There's a ventral midline, but they cannot stay. They have to go to the side. So we'll talk about that probably next few slides, probably on Wednesday.
Okay, thank you.
Yeah. Okay, so any further question that I need to clarify? Okay, then I'll just stop today and then I'll meet you on Wednesday. Thank you.
Thank you.